of the Black and Neuro seminar series for 2022-2023 year. As we start, I wanted to point out that automatic closed captioning is available. You can access this by clicking the three dots in the bottom right corner of your screen. And if you experience any issues accessing the captions or interpreters, please message Tier in the chat. She'll be ghost moderating this session to make sure everything goes smoothly for us. And before we begin, I would like to acknowledge the Gabrielino and Tongva peoples as the traditional land caretakers of Stavanger, where I am joining you from today. We're all joining this event from places around the world, and so I invite you to take a moment to reflect on the lands you are joining from, as well as your relationship to those lands and to the indigenous peoples who have and continue to inhabit them. So let me introduce myself. My name is Casey Dieters, and my role at Black and Neuro is program lead for the seminar series. And again, I want to welcome everyone to our very first talk of the year. In this series, we plan to feature neuroscience faculty, neurologists, postdocs, and graduate students. We hope to provide a stage for them to share their science, their path, their setbacks, and their triumphs. And we hope through this we can celebrate our community and help change the broader community's idea of what a neuroscientist or neurologist looks like. Some logistics after the talk ends. We'll have about 10 to 15 minutes for questions from the audience, and you can ask them using the Zoom's Q&A function at the bottom of your screen. Feel free to share those throughout the talk, and I'll be moderating the questions. Additionally, if you want to share any tidbits you learned today on social media, please do so using our hashtag BIN seminar series. Now, for some background on our speaker, Dr. Keenan Walker. Dr. Keenan Walker is an investigator at the National Institute on Aging Intramural Research Program and chief of the Multimodal Imaging of Neurodegenerative Disease, or MIND unit. Dr. Walker earned his PhD in clinical psychology from St. John's University. He completed his pre-doctoral internship in clinical neuropsychology at the University of California, San Diego and VA San Diego healthcare system before beginning an NIH-funded postdoctoral fellowship at Johns Hopkins University School of Medicine in age-related cognitive disorders. After receiving an NIH-funded K-23 Career Development Award, he transitioned to the role of Assistant Professor of Neurology at Johns Hopkins. In 2020, he joined the NIA's Laboratory of Behavioral Neuroscience as a tenure-track investigator. Dr. Walker's current research program focuses on understanding the role of abnormal immune function in Alzheimer's disease and late-life cognitive decline. Today, Dr. Walker will be talking about using proteomics to understand the peripheral drivers of neurodegenerative disease. And with that, I would like to turn the floor to our speaker. Thank you, Dr. Walker.
Thank you, Dr. Dieters. I really appreciate the introduction, and it's truly an honor to be invited to give the inaugural seminar for Black and Neuro. So, as Dr. Dieters mentioned, I'm going to be talking about using proteomics to understand peripheral drivers of neurodegenerative disease. And I'll talk more about what that means. So just first, here are my disclosures. And so we're all on the same page. I want to first, just introduce the term dementia and Alzheimer's disease. So we're talking from a common understanding. So we think of dementia as this umbrella term for cognitive impairment that is severe enough to actually interfere with everyday life. And within dementia, we have multiple different etiologies or causes of dementia. Alzheimer's disease is just one of many forms of dementia, but it is the most common. So it affects, about 2/3 of individuals with dementia, but is, like I said, comes in, dementia can come in other forms such as dementia with Lewy bodies, vascular dementia, et cetera, and they're not mutually exclusive. But Alzheimer's disease affects about 6 million individuals over 65 in the US, over 10% of the population over 65. And those who are relatively young, between 65 and 74, make up 1/3 of the population living with Alzheimer's disease. So it affects a pretty significant portion of individuals' lives. So the prevalence of Alzheimer's disease is really expected to increase over the next few decades. And right now it's at 6 million within the United States. By 2060, it's expected to be almost 14 million. So this is, we are unfortunately expected to see some massive increases in the prevalence of Alzheimer's disease. And a lot of this is due to this increase in people increase in dementia among people who are 85 and older, owing largely to the improvement in public health and medicine that's allowing people to live to this period. So one of the important things that I want to note, very germane to this seminar, is that dementia, and not Alzheimer's disease specifically, but dementia more broadly, disproportionately affects Black and Hispanic individuals. You can see here the prevalence rates across the last decade or two, and you see prevalence about 20%. These figures are somewhat old, but the prevalence rate is over double for Black and Hispanic individuals compared to those of European ancestry. So this is really it's a large public health problem, and it's an even larger public health problem for minoritized individuals. So now I'm going to talk about the clinical and pathological features of Alzheimer's disease. And when I say clinical, I mean the symptoms that patients express, and what Alzheimer's disease is typically characterized and how it's differentiated from other forms of dementia is this pattern which typically consists of memory loss, like an amnestic memory loss or forgetfulness. And often with that is word finding difficulty, trouble remembering names, for example. There's also psychiatric symptoms such as depression, anxiety, and apathy are common, but also you see behavioral changes. But Alzheimer's disease is more and more trying to move in a direction of being diagnosed pathologically. And previously, this was only done at autopsy, but now there's ways to do it in vivo. But pathologically, Alzheimer's disease can be defined by the appearance of plaques and tangles. And those are really the key features. And when I say plaques, I mean these protein fragments or peptides defined, we'll call amyloid beta, that are clumped together outside the neuron. And then we have the tangles, which are tau neurofibrillary tangles, these microtubulin proteins that become hyperphosphorylated within the neuron and basically impair the microtubule or neuron's ability to transmit information in organelles and other important molecules. So these are the core features, plaques and tangles. But Alzheimer's disease, consists of other pathological processes that may, promote the disease even further or more aggressively. So you often see vascular dementia or vascular changes, supravascular changes in people who have co-occurring Alzheimer's disease, and things like neuroinflammation. So while plaques and tangles are key features, they're certainly not the only thing. Now, Alzheimer's disease is a very insidious process. It takes place over multiple decades. And 2 to 3 decades is what's typically discussed. But the important thing is that there is this preclinical period where people have no symptoms, and yet they have pathology on the brain, right? And the pathology I'm talking about is the amyloid and the tau and other forms of pathology that can take place that are not necessarily specific to Alzheimer's disease, but can move it along more quickly. Now, this preclinical period can last for 2 decades, or even longer potentially, but then eventually people move into the stage of what's called mild cognitive impairment, where people have, mild cognitive symptoms that are, still somewhat problematic, but not problematic enough to interfere with everyday activities. And then people progress from mild cognitive impairment to eventually to dementia. And that has multiple levels of severity as well. So a key component of my research is this idea that systemic health conditions, that are conditions that occur outside the central nervous system, can actually influence one's risk for dementia, will influence one's risk for Alzheimer's disease. And epidemiologic studies and translational research has shown that a lot of these non-central nervous system conditions, things like diabetes, hypertension, inflammation, and autoimmune conditions, can actually augment the risk for dementia. Now, this figure on the right was published by The Lancet in 2017, but the idea that I want to get across is that anywhere from 30 to 40% of the attributable risk for dementia is due largely to conditions that lie outside the central nervous system that are potentially modifiable. So we hypothesize that plasma proteins circulating, protein circulating, in the circulation, might actually mediate this relationship between systemic disease, peripheral health conditions, and Alzheimer's disease and dementia risk more broadly. And the framework we're putting forth is provided up top, where we have clinical conditions, subclinical disease, or even non-disease processes like cellular aging and cellular senescence can actually change the protein expression, in various organs and tissues outside the central nervous system. And these proteins can then, through various conduits listed here, actually influence cells within the brain to change behavior and the structure of the brain itself. Now, some of these proteins have been identified already. A lot of them are cytokines and chemokines, which are in circulation that can affect target cells within the brain. We propose that there's many, many more proteins that are peripherally circulating, so systemic, that are involved in Alzheimer's disease and dementia risk. And really, that's what we've aimed to do over the last 5 or so years in my lab, is really to identify some of these protein mediators and understand how peripheral disease can affect Alzheimer's disease risk in this process. And more recently, we've taken the approach of using what's called untargeted proteomics. So what we're doing is measuring large numbers of proteins in the plasma or in the serum. And most commonly, we're using a platform called SomaScan by the company SomaLogic. But we're able to measure anywhere from 5,000 to 7,000 proteins in the plasma with a fairly small amount of blood, and then in an unbiased way relate these proteins to outcomes of interest, namely dementia risk and other CNS related or neurocognitive outcomes. Now, I like it. It's a data-driven approach. It doesn't require a prior hypothesis, and we've been able to generate some really interesting data from using this untargeted approach. Now, we published a paper last year in which we, in a set of older adults, identified a number of plasma proteins that were associated with dementia risk across a 5-year follow-up period. And in addition to identifying a number of dementia-associated proteins, we actually were able to show that some may, in fact, be causally linked to Alzheimer's disease. But one of the big issues with this study particularly, and a lot of the proteomic work that's been done to date, is that they really focus on older adults. And we know that, as I mentioned before, Alzheimer's disease and dementia more broadly has a very protracted preclinical period, a preclinical period extending 1 to 2 decades. And the consensus now is that pathogenesis for Alzheimer's disease, for vascular dementia, for other forms of dementia, it begins in middle adulthood, if not earlier. So in order to really understand the early biological features of dementia, Alzheimer's disease, vascular dementia, et cetera, we really need to look at midlife. And that's what we've done. So we've conducted a midlife plasma proteomic analysis of dementia risk across the subsequent 25-year period. And so our first objective was really to understand the early peripheral biological changes associated with future Alzheimer's disease and dementia more broadly. And secondly, we wanted to identify novel biomarkers in plasma that we could use to stratify people for the specific risk for Alzheimer's disease, dementia, and other relevant phenotypes. So to do this, this analysis was conducted in several steps. And the first step involved discovery, and this was done in the ERIC cohort. So the ERIC cohort is the atherosclerosis risk in communities cohort. It's a multi-site community-based study done within the United States. It started in the late 80s. But what we did was we took blood from the 3rd visit of this longitudinal cohort, and we measured about 5,000 proteins in 11,000 individuals. And we related proteins one by one to dementia risk over a 25-year follow-up period. So over this 25-year follow-up period, about 1,900 people developed dementia, and we looked one by one at those protein dementia risk associations over this period. We also looked at what's called near-term and long-term dementia risk, which I'll explain in a second. So here are the results from our first discovery analysis. And like I said, we looked at 5,000 proteins in relation to dementia risk over 25 years. The volcano plot on the right shows, very simplistically, the results of this analysis. So this represents 25 no, 5,000 associations, still a lot. But what we see is a GDF15 was our top proteins, rising well above the others in terms of its significance. And we saw 25 others that were associated with 25 other unique proteins associated with future dementia risk. And I'll mention that this is with adjustment for demographic characteristics, cardiovascular risk factors, kidney function, so these potential confounders. And so we think these proteins are fairly independently associated with future dementia risk. And this is with multiple comparison adjustment at a pretty conservative level. So we did the same thing, but this time we looked at what's called near-term dementia risk. We looked at the 5,000 proteins in relation to dementia risk, but over a span of 15 years. So measured proteins at midlife, looked at 15 years outward, and we saw this time 6 or 7 proteins that are associated with dementia risk. We again saw GDF15, but we saw a few others that were not identified in the full follow-up analysis. So in that initial analysis. So we have this unique set of proteins associated with dementia risk in the near term that are likely more relevant when pathology is already present on the brain. They may accelerate the dementia process or the pathological process underlying it. We're not quite sure yet, but this is a distinct set of proteins linked to near-term dementia risk. And then we looked at proteins at midlife in relation to now long-term dementia risk. And this, as you might expect, is dementia beyond 15 years. So these proteins weren't unique. They were identified in our full-term analysis, but what we can say about these is that they're differentially expressed or perturbed 2 or more decades before the onset of dementia. So probably involved in some of the earliest features of the process, whether it be vascular changes, amyloidosis, or immunity. So we put everything together. We have, here's the Venn diagram. We have GDF15, which showed up in all analysis, and we have a set of proteins that are distinct, that are associated with near-term dementia risk. Now, a number of the proteins, those with stars next to them, have been nominated by the Accelerated Medicine Partnership, or AMP-AD, or AMP Alzheimer's disease, as potential therapeutic targets. And the AMP-AD consortium really looks at brain tissue proteomics to generate their therapeutic targets. So we're seeing this convergence between plasma proteomics and brain tissue proteomics in terms of nomination of important proteins. But what we did next was we wanted to ask the question, well, when we measured proteins in midlife, the average age was 60. So, although I consider that midlife, it's probably on the later end of midlife. And we wanted to know if we really did have bona fide midlife biomarkers. So what we did was we cut the sample in half, and we looked at these proteins again in relation to dementia risk, but this time just looking at the younger half, so people 60 or younger than 60. So in our 40 and 50-year-olds, we have the analysis for the 40 and 50-year-olds is in red. The primary analysis that I showed you already is in blue, and the hazard ratios are plotted on the x-axis. And what you can see is that the associations are either very similar or even in some cases stronger when we look at 40 and 50-year-olds exclusively. And so my takeaway from this is that we indeed have some midlife biomarkers, some proteins measurable in 40-year-olds that may give us some telltale signs of dementia risk in subsequent decades. So we can then look at the biology of our proteins. We're now looking at our full set of 32 nominated proteins, and we see that they fall, most of them, into one or more biologic categories, right? We have immunity, we have proteostasis, and protein regulation, we have synaptic function, we have extracellular matrix organization and degradation, vascular function, and metabolism. And we see these biologic things highlighted by our proteins, but we can also use a more comprehensive approach to understand the biology. And this is sort of an enrichment analysis. And what we're doing here is looking at the overlap between our set of proteins and the set of proteins known to exist in specific biologic pathways that have been curated and annotated for us. So we extended our scope to look at all the proteins that met a less stringent threshold. So we're looking at, for example, the top 200 or 300 proteins in this analysis, but we see more overlap than we would expect by chance for a lot of different biologic pathways. And those are the ones that are shown here above the dotted line. So when we look at what's called near-term dementia, we see a number of interesting biologic pathways. I just highlight some of the ones I'm personally interested in with the red arrow, but everything that's green is predicted to be an activated pathway. Everything that's blue is predicted to be an inhibited pathway, whereas the gray, the activation either, usually could not be predicted. But we see a lot of activated biologic pathways in the 15 years leading up to dementia risk. And we see pathways such as platelet-derived growth factor, so vascular parasite function, right? We have interleukin-3, JAK-STAT signaling, were highlighted here, which suggests immune activation. Now, if we look at earlier in the process, so 15 years onward, we see a much different pattern, right? We see enrichment for matrix metalloproteases, unfolded protein response, some more proteostasis and extracellular matrix organization. But we also see bile acid biosynthesis and granulocyte and adhesion diapedesis. So, some immunity, some bile acid biosynthesis nominated there and not necessarily activated. So we get a sense of the biology from our enrichment analysis, but we also wanted to see whether or not our nominated proteins replicated in external cohorts. ERIC is a big cohort and it's a population study, but we still want to ensure that the results we see are generalizable. So we worked with 2 additional cohorts, the EMIF study and the Whitehall 2 study. And I'm going to talk about some of our work in the EMIF study with our collaborators. So in this study, this was done in the United Kingdom, it's European Medical Information Framework. We were able to look at our set of 32 proteins and see if they were at different levels, so differentially expressed in people who were clinically diagnosed with Alzheimer's disease compared to those who were clinically assessed to be cognitively normal. And you can see in the heat map on the right, those proteins that are in gray weren't measured in this replication cohort, but we can see for those that were measured, many of them are differentially expressed in Alzheimer's disease patients versus controls. Okay, so this was some reassuring evidence that what we were seeing at midlife replicated outside of our cohort and was relevant potentially later in life when people actually had clinically defined Alzheimer's disease. We also found that a lot of our proteins were associated with progression from that pre-dementia stage, the prodromal stage of mild cognitive impairment, to future Alzheimer's disease. So, again, more evidence of their potential for prognostication and risk prediction. So using this cohort, we were also able to look at whether our proteins were neurobiologically relevant from a more molecular perspective using CSF data. So using cerebral spinal fluid, we could relate, cerebral spinal fluid biomarkers, we can measure our proteins in plasma and see if their levels related to CSF biomarkers of amyloid, tau, and in other analytes that I'm going to talk about here. But first, so we looked at amyloid and phosphorylated tau as our Alzheimer's specific biomarkers, and we see in the heat map, these first 2 rows, that a number of our proteins that were measured are associated with amyloid positivity and elevated phosphorylated tau. So what this tells me is that these proteins are relevant to Alzheimer's disease. Now, we also see that many of these same proteins are associated with total tau, which is typically an indicator of just, non-specific neurodegeneration. But we also looked at other neurodegenerative markers as well. And we see neurofilament light, which is broadly used as a marker of neuronal injury. So a lot of our proteins in plasma, again, are associated with neurofilament light in CSF. And then skipping down to YKL-40, which is our marker of neuroinflammation, again, measured in CSF, we see that this neuroinflammation marker is again associated with some of our plasma dementia-associated proteins. And I'll call out GDF15 here, which was our top dementia-associated protein in the ERIC study. And we saw that it was also associated with Alzheimer's disease, also associated with cognitive decline in the Whitehall 2 study. And I didn't show that data, but it seems to be a very strong risk factor for clinical progression. And what we see here is that it's not associated with amyloid or phospho-tau or neurodegeneration, but seems to be related to neuroinflammation, suggesting that this GDF15 protein might not be Alzheimer's specific, but maybe more broadly linked to a process that cross spans numerous neurodegenerative conditions, potentially neuroinflammation, as shown by this correlation here. So next, what we did was we asked the question, okay, we have our list of proteins, we're gaining a little bit more confidence in them. Where do they come from? And we were able to use the GTEx and the Human Protein Atlas databases to answer this question. And here are these results. The gene expression data is on the left, the protein expression data is on the right. And there really are three takeaways, I would say. So the first is that there's a number of proteins that are just expressed broadly across tissues that don't seem to be nonspecific. So when we pick them up in plasma, where they come from is anyone's guess, despite the fact that their levels still relate to our neurocognitive outcomes. We have the second set of proteins that are specific to the central nervous system, to the brain specifically, and most of those ended up being synaptic proteins. We have proteins here like complexin 2, complexin 1, somewhere on this heat map, that seem to be expressed primarily in brain tissue when they are. So when we measure them in plasma, we think this is a derivative of an indicator of something that's happening in the CNS, make for good biomarkers. And then we have these proteins that aren't expressed in brain tissue at all, yet they are strongly associated with dementia risk, strongly associated with some of these other endophenotypes. And that's the case for GDF15, which is not detectable in brain, yet associated with neurocognitive outcomes very strongly. So those proteins that were detectable in brain, what we were able to do was use existing data that's out there and see if their levels in brain tissue are different in the brains of people who died with Alzheimer's disease and those who passed away without Alzheimer's disease. So we're comparing 80 brains to control brains, and we see for those proteins, many of them that were measured and detectable in brain are differentially expressed. So we see our complexin 1, complexin 2, cerebellum 4, our three synaptic proteins are downregulated in the AD brain, and then our heat shock proteins and epidural growth factor receptor implicated in immunity, these are upregulated in Alzheimer's disease brains. So, we have a set of midlife dementia-associated proteins, like identified in 40 and 50 and 60-year-old in plasma that show a pretty good amount of overlap with differentially expressed proteins in Alzheimer's disease brains of people much older. And so different tissue is an important sort of takeaway there. So we also used the network approach, and this was very informative in terms of going back to understanding what the biology is telling us. And for this, I worked with Pascal Schlosser and Linda Zell, pictured here at Johns Hopkins. And what we did was we used a program called NetBoost. This is a data-driven clustering program, which brings together proteins into modules or clusters based on co-expression. And so we found that when we did this, we applied this to all 5,000 proteins measured in middle-aged adults. And we found 19 protein modules were extracted. And these protein modules can be seen in the dendrogram below and are also expressed in the heat map in the bottom right. But these protein modules and modules of co-expressed proteins are mutually exclusive. We found modules ranged in size from 20 proteins to 2,000. And we could quantify person-specific expression. And so with that, we can calculate the degree to which each person expresses each of the 19 modules, and then we can relate module expression to dementia risk. And that's exactly what we did here. And so again, these are midlife protein modules in relation to dementia risk over 25 years. We have, after adjustment for confounders and multiple comparison correction, we found 3 modules, 1, 5, and 19, that were associated with dementia risk. Now, we did the same thing for near-term dementia risk within 15 years and found module 9, right? So, different follow-up period, we find a different module that's associated with the outcome of dementia. And then we look at our long-term dementia risk, okay, 15 years outward, and we see 1, 5, and 19. So it looked, again, a lot like our full-term follow-up here. So then, these modules are arbitrarily named, but, insight only comes from understanding what the modules represent. So we looked at module 1, and this is a big module, 700 proteins, but we were able to, again, do an overrepresentation enrichment analysis to understand what the big set of proteins really represented from a biologic perspective. And the takeaway here is that it's very much an immune-related module, right? We see an enrichment for JAK-STAT signaling, TH1 and TH2, so T cell signaling, a toll-like receptor ligation. So there's other biologic processes here too, but the overall takeaway is that this is an immune-based module. The protein loadings on the individual protein loadings on the module are shown on the right, just for the top 50. But we did the same thing for module 5, somewhat smaller, about 200 proteins. This one was an extracellular matrix assembly or degradation module, I would say. Some immune signature here, but mostly extracellular matrix organization. We look at 19, again, another dementia-associated module. It's weakly enriched for immunity, also MAP kinase signaling, as you see here. But if you look at the hub proteins, which are just sort of the top 6 proteins picked arbitrarily, 6 is an arbitrary number, but these are the proteins most strongly linked to overall module expression. And here it's very informative. We have 3 heat shock proteins listed among our hub proteins, which suggests to me that this is like really a stress response module, and hence the immunity that we're seeing listed here as well. But the asterisks here show that, and they represent the fact that a lot of these proteins are differentially expressed at an RNA level in the Alzheimer's disease brains as well. So we looked at the hub proteins and saw, hey, these are relevant for Alzheimer's disease in brain tissue and are relevant for a midlife plasma protein module that predicts dementia risk over 25 years. So we think this is a really important one, module 19. And then module 9, which is our near-term dementia-associated module here, really enriched for complement and coagulation. So different biology here, and again, very much enriched for Alzheimer's related proteins, at least when we look at the RNA level. So if we put all these pieces together and try to understand the directionality, and the temporality, we can gain some insights. And what a lot of the modules were immunologically related. And this is a question that I've really been thinking about for a fair amount of my relatively short career, is how does peripheral immunity influence Alzheimer's disease risk? And the findings have sometimes said it's inflammation, immune activation is good, sometimes it's bad. But this paints a very complicated and dynamic picture. But I think we're getting closer to understanding why we're seeing mixed results. We see downregulation of JAK-STAT signaling, for example, associated with future dementia risk. At the same time, upregulation of leukocyte activation is associated with dementia risk. So this underscores this idea that immunity is complex, and we see these different arms of immunity being either affected or affecting dementia risk. Now, as we get closer to the dementia onset, when we look at module 9, we see more complement and coagulation coming into the picture here. So we see this dysregulation in another, albeit related system. So next question we wanted to understand or ask is whether or not the proteins and the protein networks that we really have been looking at so far are causally linked to Alzheimer's disease. They're associated, that's clear, but are they mechanistically involved? And we were able to do this with help from Marion Fornage and Yeonju Yang from UT Health Science Center. And what they did was they performed a genome-wide association study on plasma protein level. So for all of our 32 dementia-associated proteins. So we found protein quantitative trait loci or variants that predicted plasma protein level. We can then take these pQTLs and put them in what's called the Mendelian randomization framework and really answer the question of causality, or at least find some evidence for it. So, this is just what we did. And for those not familiar with Mendelian randomization, it really uses this random allocation of alleles in the population to sort of make these causal inferences and do away with or downplay the issues of confounding, issues of reverse causation that plague a lot of the epidemiologic studies. And so the bottom line here is that when we looked at whether proteins were causally linked to Alzheimer's disease, with this genetic approach, we found that calcitonin 3 and serpinate 3 showed some evidence for a causal link. Now, there's a lot of sensitivity analysis that are done to assess the Mendelian randomization assumptions, and actually serpinate 3 was robust to many, if not all, of those sensitivity analysis. And so we have some confidence that this is actually mechanistically relevant, whereas calcitonin 3 wasn't as robust. This is a really interesting immunologically related protein. It's an acute phase protein involved in some interesting processes. So we looked the other way too. We asked and answered the question of whether or not Alzheimer's disease itself is actually causing changes in protein level. And that's why we're seeing this differential expression. So when we looked in the reverse direction, we see, a lot of our proteins are actually predicted to be differentially expressed as a result of Alzheimer's disease. So I think this is interesting because we found these proteins were differentially expressed in 40 and 50-year-olds. And this suggests that the Alzheimer's disease biology or Alzheimer's disease genetics may already be having effects on systemic biology, protein expression, in midlife. But one of the other takeaways here is we have a big set of dementia-associated biomarkers, Alzheimer's related biomarkers that have been genetically validated at this point. So, pulling all this together, we now have this new set of plasma biomarkers for Alzheimer's disease and dementia that, have been linked to distinct pathways. Now, these are proteins that can be measured in 40 and 50-year-olds that might represent breakdowns in different aspects of biology relevant to dementia risk, relevant to Alzheimer's disease. Proteostasis and immunity came out big here, and the levels of evidence I put next to each protein. We also have a bunch of pretty novel biomarkers for synaptic function or dysfunction and extracellular matrix organization and vascular function. So wrapping that story up, I'm going to switch gears a little, but come back to this theme that plasma proteins, we think, mediate this relationship between systemic disease and brain changes that underlie Alzheimer's disease. And the second theme here is we can use proteins in blood to really understand the effects of systemic diseases that we're all very familiar with on brain function. And to illustrate this, I'm going to talk about some work that was done by one of my postdocs, Heather, dark pictured here. And what Heather did was she related she used protein indicators of health to understand how peripheral health may affect Alzheimer's disease as measured by a number of biomarkers. And let me tell you what that means. So she used what's called a soma signal test. So these are protein-based health indicators. And they are, again, protein-based indicators of like health, disease risk, conditions, or traits that are listed down here. And how these protein-based health indicators were developed was measuring thousands of proteins on thousands of patients and using a machine learning approach to find the best combination of proteins that predict the clinical outcome. Now, we then, using the same set of proteomic measurement in our cohorts, here we use the Baltimore Longitudinal Study of Aging, we can then measure derive the same protein-based health indicators in our cohort of people who are cognitively normal and fairly healthy, and relate these protein-based health indicators to previously measured Alzheimer's disease and neurodegenerative biomarkers. So here we have our amyloid and tau. We also have our neuronal injury and astrogliosis biomarker here measured from plasma. But we could also relate each of these protein-based health indicators to brain volume and molecular changes on the brain as measured with PET scan. So here's some of Heather's results, and the overall takeaway is that she did find a lot of evidence to suggest that protein indicators of health do relate to a number of our plasma neurodegenerative biomarkers. So again, we have Alzheimer's disease biomarkers, first two columns, and then our neuronal injury and astrogliosis on the third and fourth column. And there's a lot of information here. These are covariate adjusted results. I'll highlight some that I found particularly interesting. But one is that as we see more lean body mass, we see higher levels, people with more lean body mass from a proteomic perspective. So they have the proteome of people with high lean body mass, I guess you can say. We see more phosphorylated tau, but less NFL, less neuronal injury, and less astrogliosis. Very interesting because these three biomarkers should sort of rise and fall together. So there's some pleiotropy here, perhaps, well, at least in terms of the relationship with lean body mass that I think could be explored. We have some evidence here that kidney function is very relevant to Alzheimer's disease and neurodegeneration, or at least the biomarker levels themselves, which is something that's becoming more appreciated. And then we look at our cardiovascular risk biomarkers down at the bottom. We see proteomic risk for primary cardiovascular disease, not so much related to our Alzheimer's and related dementia biomarkers, but proteomic risk for secondary cardiovascular disease, meaning you have the proteome of someone who's at risk for having their second myocardial infarction or stroke. If you have that proteomic signature, you also have elevated levels of these ADRD, Alzheimer's and related dementia biomarkers. We also have this HFpEF and HFrEF. There's a heart failure with preserved ejection fraction, heart failure with reduced ejection fraction, prognosis biomarkers. And although we don't have people really with heart failure in our cohort, we find that if you have the proteome of people who have poor prognosis in the context of heart failure, you have high levels of these Alzheimer's and related dementia biomarkers. So, just to make it clear, we're looking at protein-based proxies of health conditions in people that may not actually have the health condition, but we're able to look at subclinical disease traits is what I would say. Now, we also looked at these protein-based health indicators in relation to brain amyloid measured on PET scan. And this is in a subset. We looked at amyloid because, again, it's a key feature of Alzheimer's disease. But we found after a covariate adjustment, one of our protein-based health indicators, this is a proteomic indicator of poor prognosis in heart failure with preserved ejection fraction, was associated with increased odds of elevated amyloid. Now, this was interesting because those of you who, this is a neuro event, but those of you who follow like the heart research, cardiac amyloid is a key driver of heart failure with preserved ejection fraction specifically. And so it's related to the amyloid that's on the brain in people with Alzheimer's disease. So what this tells me is that the proteomic signature of people with cardiac amyloidosis and at risk for heart failure might be similar to the proteomic signature of people with brain amyloidosis who are at risk for Alzheimer's disease. Now we took the set of people with brain amyloid and we separated them into people who are amyloid positive and thus on the Alzheimer's disease trajectory somewhere, and those who are amyloid negative and thus likely not on the Alzheimer's disease trajectory. And we found that primary and secondary cardiovascular risk and our heart failure with preserved ejection fraction biomarkers were associated with elevated amyloid in people on the Alzheimer's disease trajectory, but not in people not on the Alzheimer's disease trajectory, which makes a lot of sense on its face. But further underscores the relevance of these cardiovascular risk factors, measured through a proteomic lens. Now, we also looked at brain volume with MRI in the BLSA cohort, and we looked at this signature called SPARE-AD, which is really just the degree to which your pattern of brain atrophy reflects the pattern of brain atrophy seen in people with Alzheimer's disease. And we see that the same cardiovascular risk factor proteomic biomarkers are associated with a pattern of Alzheimer's, appearing atrophy, as indicated by these scatter plots here. Some more evidence for the relevance of these biomarkers, given the link between these protein-based biomarkers and atrophy, we wanted to look to see if these molecular changes that we can measure in blood might actually mediate these relationships. And we did find some evidence for that. And we then took these same protein-based health indicators, we computed them in ERIC because they also have the same proteins measured. And we asked the question, well, if someone has a proteome of someone with primary cardiovascular risk or secondary cardiovascular risk, what is their risk for future dementia over this 25-year period? And as you can see with the green line, those with primary and secondary cardiovascular risk had much greater risk for future dementia. And the same was the case for people with proteome of people with poor prognosis in heart failure. Again, not people with heart failure, but people with a proteome of poor prognosis of people with heart failure. I know it's a little bit to wrap the head around, but really we're able to look at these proteomic indicators of diseases that may not even be there, may never be there, but people can still express some clinical traits of these disease patterns. And so we think we have more sensitivity using this proteomic perspective. So just the takeaway from this is that we can use these proteomic health indicators to understand how peripheral disease may influence neurodegenerative biomarkers, right? These things are becoming super popular, even entering into the clinical realm. And I think it's important that we understand how comorbidity, how health, how physical traits can affect individuals' levels before we, go forward in diagnosing people based on these biomarkers or recommending treatments. And then this also, helps us understand how peripheral health can directly affect the brain, because we see some convergence between our plasma biomarkers and our neuroimaging biomarkers. So we think, we're really, with this, capturing and better understanding how things like cardiovascular disease in people who are not expressing it clinically might affect Alzheimer's and neurodegenerative related outcomes. And with that, I'd like to acknowledge, all the people who made this work possible, especially people in my lab at NIA and the collaborators, many of which come from the ERIC cohort, mentors and past mentors, and then our collaborators who have allowed us to use their data to help support some of our findings. And then, of course, our funding sources, without which this wouldn't be possible. And then again, I want to thank all of you for listening, and thank you, Casey, and the rest of the team for the invitation to speak today. So hopefully there's time for questions.
Wow, thank you so much for that amazing talk, Dr. Walker. It really seems like your lab is doing amazing, great, cutting edge science, and the use of peripheral markers, something's not as invasive, as CSF is, I know, going to be very crucial to the field moving forward. I'd like to remind everyone that are participating in this webinar to insert any questions you have in the Q&A box below. And we have at least one, so we'll start with that. And the first question is from Christina. Such a great talk. Thank you, Dr. Walker. Given the disparities we see across race, ethnicity in terms of systemic health conditions and risk conversion to dementia, largely due to systemic factors like social determinants of health, do you expect to see differences in proteomic health across groups? And how would you recommend investigating this in the future? Very good question, and something that I was actually going to ask you as well.
Yeah, very, very good question. I mean, so I think the point that you're getting at, and the point that I hope, well, I didn't convey explicitly, but it's very important, is that these conditions, especially the cardiovascular conditions that we show to be associated with Alzheimer's disease, dementia risk, and these biomarkers that we're looking at are much more prevalent in Black and Hispanic individuals. So the prevalence of these conditions increases. That means the risk and prevalence of the conditions that they can help drive, like Alzheimer's disease and dementia, also increases. Now, whether or not the proteome is actually different between Black and white, or between white and any other race of individual in terms of detecting these conditions, we haven't yet done. And I think it's similar to like the polygenic risk score issues. And I think that's what it's going to come to, where we're going to need to build larger samples and look specifically in, for example, Black individuals to sort of recompute these protein-based risk scores and recompute the dementia-associated proteins in those samples. I think there will be, in the area cohort, we've been able to look, we've run stratified analysis, and we do, see a lot of the dementia-associated proteins that we see overall are also associated with dementia in our Black-only population and in our white-only population. So there is at least some overlap there. But I think for things like these protein-based health indicators, if we were able to recompute them in different populations, they would be even more accurate.
Yeah, absolutely. Do you think, what are your thoughts regarding there was like a little sub question there about social determinants of health. Would you expect those to impact the peripheral markers, especially ones such as like inflammation?
Yeah, I really do. And I know some great work underway on this question specifically. So I won't divulge too much because it's very early phase, but as you can imagine, people are very interested in seeing how social determinants of health affect the proteome, and how the proteome, then influences the emergence of disease, especially, things that might potentially be causal. So yeah, like you said, Casey, we know, for example, stress, discrimination, loneliness, these things can influence, for example, the immune proteome, right? The inflammation, for example, monocyte activation. And so I would expect those same sort of signatures to be reflected when people apply large-scale proteomics to social determinants of health. So there's a ton of really interesting work that I think that's going to come out in the next decade on this topic specifically.
That's great. I think in a lot of your work, I was really interested to see, and I mean, I've been seeing this in the field quite a bit, but it hasn't been my particular focus yet. And I'm starting to realize as I start my lab at UCLA, trying to think about what target population should I really be focused on. And a lot of your research suggested that midlife is pretty crucial in identifying some of these proteins. I was wondering what your thoughts are about the field. And I think like a lot of the studies that we primarily focus on are 65 and older.
Yeah.
So yeah.
Yeah, no, it's an important point. A lot of the studies are 65 and older. So, and I think there's two age groups that have been understudied. I think the midlife group is one of them, but people have caught on, and thankfully, and now cohorts are starting with enrollment of 40-year-olds, 50-year-olds, and it's a long investment, right? You don't really get a return. You can do some stuff with just 40-year-olds, but you don't get a return until they start to develop the outcomes. So it's a that's a drawback of recruiting very young, because like you don't know if someone's going to develop dementia until you wait and see if it happens. But it's definitely worthwhile research, and a lot of the good work is coming from the retrospective analysis, where we have we know their dementia outcome, and we can look back. People find banked plasma somewhere, or people find information that can link risk factors to future dementia risk in people who've already developed dementia. But I think then the oldest old is another cohort that I think more work needs to be done. And people over 85, there's much less there. And I think the risk factors for this oldest old group that is growing, and as I showed in the early slides, those risk factors are going to be different than people in their 70s, in their 60s, in their 80s. So I think really those are the two ways that we can fill in some of the research gaps: midlife and oldest old.
Absolutely. And we have another question in the Q&A from Ola. This was a brilliant talk. Thank you. You mentioned that diagnosis of dementia is moving towards a more pathology-based approach, but I was wondering if you've seen a link between any of the peripheral biomarkers you've identified and cognition in your research.
Yeah, absolutely. And we're working on relating the peripheral biomarkers to cognitive decline right now.
Mhm.
But I will say, and I had to take this slide out for time, but what we did was when we found a marker of interest, we were able to take it to the Whitehall 2 study, which is a UK-based study that with 20 years of follow-up, and it's unique because they also have midlife proteins measured. And they have a really nice longitudinal cognitive exam. And we were able to show that the same midlife proteins, at least some of them, were associated with progression or cognitive decline over time as sort of another validation effort. But yeah, I mean, the same proteins are associated with dementia. I expect to be associated with longitudinal cognitive decline. But it would be really interesting to see what the like domain specificity is, whether it's memory loss or slowed processing speed. I think that's definitely a future direction for us. But yeah, I don't think the biomarkers will ever replace cognitive assessment, but I think it may really makes a good pairing.
Excellent. And we got a few minutes left, so I'll ask another question. In thinking about translation to interventions, how do you see this work playing out for clinical interventions in the future? What would you recommend to your own patients?
Yeah. So I wish, so I don't want to get too far out over the skis and make any clinical recommendation from the stuff we're doing, because I don't think we're there yet. I mean, a goal of our work is to identify potential targets that can be targeted peripherally. I think the work I presented in the latter half of the study suggests that some of the health condition, cardiovascular risk factors, likely, much like we already know and has been shown, are continue to be risk factors for Alzheimer's and related dementia. So preserving cardiovascular health, specifically in midlife, diet, exercise, all the things we already know that are helpful for a lot of our systems are going to be helpful for our brain. That's not, that's something that's pretty widely known, but not always implemented by the population. But yeah, I think it's going to be some time, before we are able to translate, findings here into interventions. But that is ultimately the goal.
Excellent. Well, I want to thank you again, Dr. Walker, for joining us. This was an amazing talk. I actually have more questions that I will email you after this. So I really want to thank you so much for your time. And I'd like to thank all the participants for joining our first talk of the seminar series. And just as a reminder, we do have a newsletter, Slack, and connect with us on social media to keep track of all of the upcoming events. And we look forward to seeing you at the next seminar and or event.